This is an event on mitochondria, so we're going to share stories from science, some interdisciplinary approaches, and also some issues concerning society and how we perceive among patient groups and among the public mitochondrial research and mitochondrial dysfunctions and disease. Mitochondria are called the powerhouses of the cell. So they are the site in the cell where the energy is produced that we need to sustain life. But mitochondria are also important for deciding how cells will die. So frequently they're also called the chief executive organelle for the cell. So we're interested to understand how mitochondria are made and how they're sustained in a functional state when cells undergo different stresses, particularly in many human diseases.
I'm Rory Edwards. I'm a PhD student in my final year. Prior to my PhD, I'd done a master's degree at the University of Glasgow, and prior to that, I was at the University of Dundee studying molecular biology. So I work on mitochondria. In particular, I look at oxidative stress within mitochondria. Oxidative stress is a type of response or a type of stress that occurs within our cells that causes damage, and it's been implicated in many different diseases, including many mitochondrial diseases. And basically, I'm looking into proteins that are responsible for detoxifying this stress, known as antioxidant proteins. It's interesting to look at the fundamental pathways that are involved in keeping us alive and understanding how these pathways can go awry in disease, and potentially looking at ways to try and improve outcomes, and also looking at ways to try and combat these diseases.
My name is Sergio Fernandez and I am from Spain originally, so I graduated from the University of Zaragoza in Spain. I have been working on mitochondria forever, ever since I graduated from university in different labs in Spain, Italy, and I had been working in Cambridge for 7 years. And I have just recently joined this lab last June to start a very interesting project about mapping reactive oxygen species, which are a byproduct of the energy that mitochondria are producing, that can be good to tell the cell what to do, but they can also be detrimental if they are in excess. So what we're gonna do is at the submitochondrial level, we're gonna map these reactive oxygen species in different physiological and pathological settings, and we will see what the effects are on the biology of mitochondria. Mitochondria are a fascinating organelle that is mainly, they are called like the powerhouses of the cell because they produce most of the energy, but they have also many different functions, and they are a metabolic hub that houses most of the metabolic pathways that you can imagine in a cell. And that they're also very relevant for disease because malfunction of mitochondria cause devastating diseases in human beings. And we are also aiming towards understanding these diseases to find an effective treatment, which is unavailable right now.
Hello, I'm Professor Kostas Tokatlidis. I'm the Cathcart Chair here at the University of Glasgow, where I moved in 2013. In my lab, we're working to understand how mitochondria respond to stresses and how they can sustain their function under particularly oxidative stress conditions that is damaging for lipids, proteins, and also for DNA. So our efforts are to understand how mitochondria can respond to these stresses, which are very frequently observed in many human diseases, and particularly neurodegenerative diseases associated with aging. So my group here at the University of Glasgow is very international, with people from many different countries. We have students, postdocs, and technicians, and we have set up a network of collaborators here in Glasgow, but also in Scotland. And we work with them in different aspects of mitochondria biology and how mitochondria biology really underpins human conditions like neurodegenerative conditions and cancer and diabetes. The event that we have organized, which is a webinar that took place on the 17th of September, really was the first of its kind that was organized in Scotland. In the context of the Global Mitochondrial Awareness Week, we brought together scientists that are working on mitochondrial research, but also social scientists, designers and patient groups, so that we can actually blend these different groups of people together that are normally not participating in conferences.
So it's a great pleasure to talk to you about the mitochondria from a chemistry perspective. Now let's go back to basics. We all think of mitochondria as the powerhouses of the cell. That is, they take the fuel in the form of food and it's broken down to metabolites that are used within the mitochondrial matrix to drive the power formation, that is the formation of ATP in the inner mitochondrial membrane.
My name is Elio Cacavale. I'm a reader in transdisciplinary design innovation at the Glasgow School of Art. I'm delighted that I was able to co-organize the Mitochondria and Us webinar. The webinar has given us a unique opportunity to bring science and society together through different expertise and life experiences. The project is based in the Innovation School at the Glasgow School of Art and explores the potential for exchanges between design and biochemical sciences.
This event has been very popular. We have had over 350 participants from all over the world, Hong Kong, India, Bangladesh, Morocco, Brazil, Iceland. We have had participants both from the disciplines that are actively working on mitochondrial research, chemists, physicists, biologists and medical doctors, but also social scientists, patient group organizations and designers.
Hi, my name is Dr. Michael Pierre Johnson and I'm an Innovation Leadership Fellow in the Creative and Digital Economy at the Glasgow School of Art. This had brought together designers and biochemical scientists just to share disciplinary expertise, perspectives and identify opportunities for future collaborations.
So my name is H Hotzko. I got my bachelor in Denmark and I just recently started working in this lab. I worked with the proteins that are inside mitochondria outside of the cells and the mitochondria, and I helped get them ready and see how they interact with each other, but I also help produce them for everyone else in the lab so that they can use it for their own experiments. And I think mitochondria are very important. They are very important parts of the cell. They're also very interesting. They're kind of their own entity, kind of the black sheeps. They kind of work a bit different than the cell. They behave like their own thing, but they are very important in regulating everything that goes around the cell.
My name is Jamie Lees and I'm a Master of Science by Research student in the Tokyo-Lidis lab. I've been here since last October, so I've been here for pretty much a year now. My main project at the moment is looking at this specific peptide known as N18 and its specific capacity to enter the mitochondria of the cell for potential future use for therapeutic targeting of drugs to mitochondria for people with mitochondrial disease. More specifically, I look at the two different halves of the N18 peptide and to see what's more involved in its structural role and what's involved in its functional role. If anything, one of the most important things that I've learned from my time in the lab is the whole concept of working in the lab, the community that surrounds it, and just all the basic skills and drills that you need that will be applied to any laboratory environment. So it's been massively rewarding. So mitochondria are massively important for any cellular function. They produce all the energy that your body needs to survive and thrive. So to be able to further understand these specific organelles and their higher roles within cells, I think that's massively important for any future scientific endeavor, really.
What's mitochondrial disease? Small, strong and full of energy, just like the mitochondria which are in every cell in your body. Mitochondria are the life force inside us all. They are the power supply in all our cells, providing energy for them to survive, thrive, and make sweet music. Our body's organs, brain, heart, liver, are high maintenance and require a lot more energy to perform their functions. When someone has mitochondrial disease, their mitochondria have become run down, and the cell's power goes out. They stop performing and start to die. So the body's organs don't have enough energy to work. Mitochondrial disease can affect anyone. Recent research shows that about 1 in 200 babies in the UK are born with genetic changes that can cause it. Faulty mitochondria have been linked to a number of common diseases. Whilst some people do not show symptoms, others develop problems that are undiagnosed or misdiagnosed. There are still very few effective treatments and no cure. For children with the disease, it can be debilitating and their lives can be cut tragically short. Here at the Lilly Foundation, we raise awareness and support families who are living with the condition or who have lost a child to mitochondrial disease. We are also working towards finding a breakthrough in mitochondrial research that will eventually lead to a cure, with the hope of saving many children's lives and children suffering in the future. There is hope. There is the Lilly Foundation. Help us make a difference.
We have been able to explore cutting-edge research in mitochondria and also discuss the more societal aspects of mitochondrial research and mitochondrial disease from the patient perspective and also from the perspective of bioethics and sociology. So these events explored crossover interactions between the scientists and the more general public, and we have been able to think of innovative ways to engage the larger public and also explore these possibilities further. Mitochondrial research underpins the primary mitochondrial diseases that are incurable, often misdiagnosed, and extremely complex to detect and also to devise therapies for. So we have been able to discuss with international experts on the cutting-edge advances using an interdisciplinary approach and how these advances can facilitate new therapies which are absolutely vital for patients. We have also been able to explore the interactions between the patient groups and the scientists and how these devastating diseases are perceived at the societal level.
And as designers, our focus is to identify opportunities for new ways of supporting people living with the condition, which can lead to impacts on the quality of life and the quality of future care experiences.
So, over the last 5 or 10 years, scientists around the world have been really coming together to try and address this problem and whether or not there is any possibilities at all of developing therapies for mitochondrial disease.
We recently have observed that cells that are lacking COQ6 or are mutated in COQ6 can benefit from treatment of vanillic acid that will restore the synthesis of coenzyme Q, which opens the door for new treatments of this type of diseases.
So what I think what we can learn from these modern microscopy techniques is that mitochondria are dynamic, they are complex, and they are very beautiful, at least in my eyes.
So as we have heard, mitochondria are very complex organelles, and identifying a mutation in a gene causing mitochondrial disease is only the first step in a long process of understanding what are the consequences of the mutation in the cell.
And we are able to increase the complexity of our analysis by experimental setups and later by complex inferences about mitochondrial function and how mitochondria can change the function of our cells, of the organs of our body.
So our mission at the Lilly Foundation is to improve the lives of those affected by the condition. And we actively pursue this in 3 key ways. So firstly, we provide essential support to patients, families and carers. Secondly, we educate and inform and raise awareness of mitochondrial disease. And thirdly, we fund scientific research.
Because the disease is rare and represents relatively small groups in each country, it made sense to organize ourselves on an international level.
It is important to note that being affected by the disorder or having lost a child from it did not necessarily mean that patients abandoned the idea of having children. Often, it was precisely what was giving them hope and purpose, or would enable them to reinstate a sense of normality in their life.
At the Glasgow School of Art, by co-design, we mean fully opening up the design process and meaningfully involving people who will be impacted by an innovation in its conception and development.
Now I turn to mitochondrial donation and some of the ethical challenges of this. One strategy for reducing the chances of mothers transmitting mitochondrial disease involves intervention in the process of family making. Although IVF is a standard treatment for infertility, its adaptation to avoiding the transmission of faulty mitochondrial genes is recognized as challenging, not only for technical but for social and ethical reasons.
We're here at the University of Glasgow, where my laboratory is based, and we work on mitochondrial biogenesis. So we try to understand how the powerhouse of the cell assembles and how it functions under different cellular conditions. Our work here is quite international, also in the sense that we are engaged with collaborations all over the world. So we collaborate with groups that are working on fundamental aspects of mitochondria research, like ourselves, and are based in Europe, in Japan, and also in the US. But also we collaborate with groups that are more focused on developing therapies for mitochondria, and we try to put all these expertise together so that we can address the problem of mitochondrial disease. In the event that we have recently organized, so a webinar that was attended by more than 350 people, we got together with the Glasgow School of Arts and the Lilly Foundation, which is the UK charity specifically focused on mitochondrial diseases. The webinar was hosted also by the Royal Society of Edinburgh, and it was a very interdisciplinary event where we actually got together with ourselves and other researchers working on mitochondria research, but also together with patient groups and sociologists, social scientists, and also designers. Our effort was to establish a crossover between these different disciplines and to elaborate on new and innovative ways of engaging with the patients and also the society more generally.